Hello everyone, InformationBoxTicket Lifestyles brings you a topic from food microbiology, Bovine Spongiform Encephalopathy. This is a fatal neurodegenerative disease that can be passed from person to person. It is brought on by proteinaceous agents and causes neuronal loss, spongiform lesions, astrogliosis, and the cessation of inflammatory responses. Due to the fact that it affects both humans and animals, particularly domestic cattle like dairy cows, it is also known as prion sickness or mad cow illness. Few points that will describe this disease are: the infectious protein is thought to be a typical host prion protein that has undergone a post-translational alteration to alter its structure. The altered protein becomes resistant to enzymatic activation and inactivation. Brain inflammation, rabies, whooping illness, and cerebral listeriosis are a few of the often caused degenerative disorders of the brain. In UK, these cases peaked at more than 300 cases per week in 1993, and since then the disease has become a major public health concern. Scabies host are sheep and goat, chronic wasting disease, deer, yak and moose, transmissible microencephalopathy, mix, feline spongiform encephalopathy, domestic cats and wild lions, pumas, cheetahs and tigers, and exotic euglottalates encephalopathy are other prion diseases categories. Fatal similar insomnia, Creutzfeldt-Jakob disease, Creutzfeldt-Jakob disease, and variant humans contract the Creutzfeldt-Jakob disease by eating certain foods. So this was the introduction of bovine spongiform encephalopathy. Now we'll look at the table of content which are included in this lecture. Sources and causes of contamination of bovine spongiform encephalopathy, pathogenic mechanism, signs and symptoms, epidemiology, diagnosis, treatment and vaccination, and lastly prevention and control measures. Let's head to the sources and causes of contamination of bovine spongiform encephalopathy. The prion, a misfold protein that causes neurological disease and is resistant to extreme heat and pressure, is the cause of this disease. The degraded protein converts an alpha helical structure into a beta sheet and then generates a short chain that kills cells. Multiple cell deaths result in lesions in the brain, which can then develop into other neurological disorders and ultimately death. When a healthy animal comes into contact with contaminated food supply, particularly polluted meat, the disease is spread. It is thought that many animals become infected after eating tainted meat and bone meal. By feeding meat and bone meal to young calves and using scabies infected sheep products, other cattle becomes affected. Variant Creutzfeldt-Jakob disease, which is thought to be transmitted to humans when they become infected, may be brought on by contaminated meat from slaughtered and diseased animals, particularly the brain, spinal cord, and digestive system. Kindly don't forget to give this video a like, subscribe to my channel, and don't forget to press the bell icon button so you don't miss any of my lectures. Now we will head to pathogenic mechanism of bovine spongiform encephalopathy. When BSE agents are consumed in sick meat and bone meal, infections can happen orally or intracerebrally. When the BSE agent enters the digestive tract, it uses specialized macromolecule transporter M cells to pass through the tonsils and gut epithelium and the mucosal barrier. The ability of the BSE agent to enter the digestive tract is also improved by proteins like ferritin and direct uptake by dendritic cells. It then gathers and multiplies in the gut-associated lymphoid tissues, primarily in the tonsil, ileum, and jejunum. The enteric nervous system, part of the gut's neural tissue, is initially infected by the BSE agent. The enteric nervous system agent gets to the brain, cervical spinal cord, cerebral cortex, cerebellum, hippocampus, and basal nuclei with the help of efferent neural pathways, which are parasympathetic and sympathetic. The infection eventually spreads across the entire brain, leading to a disorder that degenerates the brain. Bovine Spongiform Encephalopathy in cattle results in recognizable lesions in brain and spinal cord that induce astrocystic hypertrophy and hyperplasia, as well as neuronal vasculation, degeneration, and loss. Kindly give your support to this channel by subscribing to my channel so that I can make more videos for you. Signs and symptoms of bovine spongiform encephalopathy. Early signs of the illness include anxiety, which are comparable to the person's cow driving habit, suffering from herdmen exhibiting behavioral changes. Neurological infections progress slowly and have an impact on mental health, as well as sensory alteration and hypersensitivity to sound and touch. Movement and posture abnormalities, low head carriage, tremor, hind limb exarchia, decreased milk production in mammals, decreased gut chewing, slow heart rate, and eventually terminal recumbency and death are all signs of abnormality. Depending on the frequency and severity of the sickness, the incubation time might range from 7 days to more than a year. By reducing environmental factors like increased stress during travel, the severity of infection can be altered. On young cattle that are under 20 months old, the BSE agent have not been found. Now let's get to know the epidemiology of bovine spongiform encephalopathy. But before that, don't forget to like this video, subscribe to my channel, and press the bell icon button so you don't miss any of my videos. So, the majority of the cases have been recorded from the United Kingdom, where there had at one point been 37,280 afflicted cattle. The rearing of cattle and the consumption of meat and bone meals then contributed to the disease spread to other European nations. Due to various techniques of rearing cattle, dairy cows are more impacted than beef cattle. As a result, after birth, dairy calves are divided and fed artificial milk. Cats, inland, gemsbok, Yorks, pumas, cheetahs, ocelots, and rhesus monkeys are just a few of the other creatures that have the disease. After the prohibition on food and feed was put into effect, the number of BSE cases decreased by 40% annually. Japan is the only country outside of Europe with 36 BSE cases that are not imported, promoting the creation of Governmental Food Safety Commission in 2003. Kindly show your support by subscribing to this channel. Diagnosis. Clinical diagnosis is 85% reliable, and microscopic examination of the brain is used to confirm illness. Prions associated with bovine spongiform encephalopathy are estimated with the help of animal bioassays. The prion protein antigen is discovered using the enzyme-linked immunosorbent test, which reacts with the antibody. Additionally, it can find anti-pathogen antibodies. To examine pathological alteration using light microscopy, histological examination of the tissue fixed in paraffin and stained with hematoxylin and eosin is performed. The presence of prion protein deposits is identified with the help of immunohistochemistry. The staining pattern of this infection is seen using secondary antibody that specifically target particular PRP epitopes. They also distinguish between distant prion strains and pre-treat the afflicted tissues. By using antibodies bound to prion proteins that divide based on their rate of migration, the Western immunoblotting approach detects the antigen. After gel electrophoresis, 3 separate bands may be seen. The highest band is deglycosylated, meaning it contains 2 sugar molecules. The middle band is monoglycosylated, and the lowest band, known as unglycosylated, contains no sugar molecules. To distinguish between isolates and strains, researchers use the unglycosylated band. Contradictory mono and deglycosylated bands are also employed to differentiate isolates, albeit interpretations varies depending on the laboratory procedures. Treatment and vaccination. Unfortunately, there are no known cures or vaccines for bovine spongiform encephalopathy at this time. Show your support to this channel by subscribing to my channel. Prevention and control measures. The prevention of bovine spongiform encephalopathy can be achieved by eliminating all meat and other products obtained from cattle from cow feed. Number 2. By limiting the import and export of beef, meat and bone meal, live cattle, and other cattle products, it can help prevent the spread of illnesses from one nation to another. The specified risk body parts such as the brain, spinal cord, tonsils, intestines, eyes, and trigeminal ganglia are safely disposed of in slaughterhouses in the US and UK. International import and export regulations are currently in place to prevent the BSE prions from getting into human food supply chain. The relevant authority must be notified when a BSE agent is discovered so they can burn the entire contaminated animal body. Lastly, laboratory personnel should adhere to safety regulations and measures when handling infectious materials, sampling, and testing. So that's it for today. Thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel, and press the bell icon button so you don't miss any of my videos. Thank you so much for watching.